بسم الله الرحمن الرحیم. پرون مونږه دا انزایم چپټر ختم کړی، نن به د بوک نویو ګوره دا مونږ ویلو کنه. recall from your previous classes that enzyme are biological catalysts which increase the rate of chemical reaction in living cell without being used in the process. Near every chemical process that take place in living things is facilitated by an enzyme. دوی دا خبره کوي چې څنګه تاسو په شاته کلاسونو کې دا خبرې کړې وي چې انزایم موږ سره بایولوژیکل کټالیست دی او هغه ما سره کیمیکل ریاکشن تیزی په لیوینګ سیل کې خو دا یوز کیږي. نو همدغه شان وګوره کیمیکل پروسس ډیټا پلیز ان لیوینګ فیسیلیټیډ بای انزایم. موږ دا خبره کولی شو چې کیمیکل پروسس چې په لیوینګ تھنګز کې څومره هم کیږي دا په انزایم سره فیسیلیټیډ کیږي. او د سم اف آل کیمیکل ریاکشن that a cell or large living cell carry out is called metabolism. Metabolism anabolism کې دا باندې پوي کړی دی. صحیح ده. Many activities in living things are controlled by metabolic pathway in which a series of interrelated steps are involved, each one of them facilitated by enzyme. چې کوم مین اکټیویټیز په دغه کې ارګانیزم کې زیاتره اکټیویټي میټابولیک پاتوی سره کنټرول کیږي او دا انټر ریلیټډ یو سیریز اف سټیپ چې دا یو بل سره تعلق کوي. انزایم سټرکچر باره ته راشي. انزایم پروټین دی. دیر فنکشن از ډیټرماینډ دیر کمپلیکس سټرکچر. صحیح ده. The reaction take place in small part of the enzyme called active site. چې انزایم اکټیو سایټ څه ته وایي هغې باره کې تاسو ما پوه کړی. While the rest of the protein act as a framework. د اکټیو سایټ نه علاوه چې نور کوم دغه یا فریم ورک دا مې هم درته ویل چې فریم ورک څه ته وایي. صحیح ده. الوسټریک سیټ چې کوم وي. The amino acid are the active site attached to the separate molecule and hold it in position while the reaction take place. This makes the enzyme specific for one reaction only, as other molecule would not fit into active site. دوی دا خبره کوي چې amino acid around the active site attached to the separate. امینو اسید چې کوم موږ سره وي که نه اکټیو سایډ نه اټیچ شي مالیکول او هولډ کی. دا مالیکول چې کوم دی دا ما سره هولډ کی په خپل پوزیشن کې چې دی په وجه ما سره انزایم څه دی دا ما سره یو ریاکشن کټالایز کوي. صحیح ده. موډ اف انزایم اکشن. نو دلته دا خبره امپورټنټ وه چې امینو اسید اراوند اکټیو سایډ اټیچ د سپریټ مالیکول سره او دا په خپل پوزیشن کې ونیسي او ریاکشن چې کوم دی هغه روان شي. موډ اف انزایم. نو ټو هایپوتیسز دی په باره کې دی. یو لاک اینډ کی ماډل دی، یو انډیوس پیټ ماډل دی. لاک اینډ کی ماډل باره کې هم موږ ډېر خبرې کړي دي خو بیا هم دا ایمل فشر راغلی په 1819 کې suggest that each enzyme had a particular shape into which the substrate fits exactly. وی په 1990 کې ایمل فشر رالو دا سجست کو چې انزایم یو پارټیکولر شیپ وي چې ای کې سبټریټ ما سره بالکل فکس پټ کیږي. This was known as lock and key hypothesis. دې ته مونږه اصل کې lock and key hypothesis ویو. According to this hypothesis, the substrate is imagined like lock and while enzyme is imagined like a key. د دې hypothesis مطابق سبټریټ مونږه داسې امیجن کولی شو، داسې تصور کولی شو، یعنی داسې یو خیال کولی شو چې سبټریټ مونږ سره یو دغه دی لاک دی او انزایم مونږ سره یو نه enzyme is imagined like key. enzyme separate مو سره separate is imagined like lock while enzyme مو سره یو چابي دی او separate یو lock دی. As one substrate and one specific key can open one specific lock, similarly a specific enzyme can break up only a specific substrate. The active site is rigid, regarded as rigid structure and doesn't modify or change during the reaction. However, later study did not support this hypothesis in all types of reaction and therefore hypothesis were modified in induced speed model. دوی دا خبره کوي او فرض کړئ تاسو یو داسې تصور وکړئ چې دا انزایم ما سره یو چابي ده او سبټریټ پکې لاک دی چې څنګه یو سپیسیفیک تالې دپاره یو سپیسیفیک چابي وي دغه شان یو سپیسیفیک ریاکشن دپاره یو سپیسیفیک انزایم به وي. او د اکټیو سایډ چې کوم وي که نه دا سبټریټ چې کوم وي دا ما سره ریجډ وي، سخت وي اکټیو سایډ او دا ما سره نه چینج کیږي چې کله ریاکشن شروع کړي، یعنی آفټر او ډیورینګ د ریاکشن دا چینج کیږي. نو خو دا دغه چې کوم دی که نه بیا دې نه پس یو بل ماډل لارو چې په وجه دا فیل شو انډیوسپیډ ماډل. The attraction of the substrate and the enzyme from an enzyme substrate complex. Attraction the substrate of enzyme or from an enzyme substrate complex by another substrate enzyme. It was originally referred to as lock and key model. The pela أول lock and key model تصور كيدل. The current theory suggests that enzyme modify molecule are in active form. Currently موږ دا وایو چې enzyme چې کوم active site وي کنه دا ما سره inactive وي. To become active they must undergo a slightly change into structure to specific accommodate the substrate. It is said in induced model. Induced model دا خبره کوي چې د active site چې کوم دی دا ما سره ان اکټیو پام کې وي او چې کله د دې سره سبسټریټ دغه کې نو دا ما سره اکټیو دغه شي اکټیو دغه درجه دي کې سلایډلی چینج اکر شي او سبسټریټ سره پکس شي. دا انزایم دی سبسټریټ په اټیک کو صحیح ده. دا ما سره انزایم سبسټریټ کمپلیکس جوړ کوم به د یو بل نه جوړ شوي دي انزایم یو خوا پروډکټ اخوا. انډیوس سپیډ ماډل دا خبره کوي ګوره دلته سبسټریټ یو شکل دی او د دغه بل دی انزایم بل دی. دلته کې سلایډلی چینج شي داسې تیره غوندې ځای به سبسټریټ لږ پک شي دلته پک شوی دی او بیا یې واپس تللی دی. Think of way you hand change shape slightly when you shake a person hand. وی چې کله ته یو کس سره لاس ملاوې که نه مختلفو کسانو لکه لاس ورکوټی نو ستا لاس دغه رابند شي نو دغه شان وجه ته څنګه ستا لاس لکه اوس یو غټ کس لاس دی نو هغې نه به زما لاس دومره تاو شي که ورکوټی مو کال مې تاو شي همدغه شان وایي یو انزایم هم ته داسې څنګه چې ستا یو ورکوټی کس سره لاس ملاو کړو لاس ته مکمل لا تاو شي نو دا مختلف د لاس دی مختلف شیپونه جوړوي. There are three ways of thinking about enzyme catalysis. They are described the same process through in different way and you should know about each of them. دلته ما سره درې وی دي چې they are described the same process through in different way and تاسو له پکار دي چې دا ووایئ. لومبنی دی reaction mechanism. Reaction mechanism څه ته وایي؟ In any chemical reaction a substrate is converted into product. په هر یو chemical reaction کې substrate ما سره product ته convert کیږي. دا ما سره څه شی دي؟ There are three way of thinking about enzyme catalysis. یعنی مطلب دا ما سره طریقې دي چې انزایم به ما سره څنګه کار کوي درې وجې طریقې دي. there may be more than one substrate دلته کېدای شي یو substrate وي more than one product more than one substrate کېدای شي more than one product کېدای شي but doesn't matter here. In enzyme catalyzed reaction the substrate must bind to the active site of the enzyme to form enzyme substrate complex and then product is released. This mechanism can show in the figure. The enzyme is then free to start. The reaction mechanism پهله دا ده چې ما سره به سبسټریټ پروډکټ ته کنورټ کیږي. دې کې به داسې کار کیږي چې انزایم به ما سره سبسټریټ سره ملاویږي او انزایم سبسټریټ کمپلیکس به جوړوي او بیا چې کله ما سره پروډکټ جوړ شي نو دا به یو بل نه پرې کیږي. لکه دا ګوره انزایم دی سبسټریټ انزایم سبسټریټ کمپلیکس اکټیو سایډ باندې بایند شوی دی. بیا چې کله ما سره پروډکټ یې جوړ کړی دی نو یو بل نه جدا شوي دي لکه د مار. بیا ما سره چې کوم دی مالیکولر جیومټري ده. The substrate molecule fit into active site. Active site کې fit شي of the enzyme molecule like a key fitting into lock. وای داسې fit شي لکه چابي په تاله کې. Once there the enzyme change shape slightly distorting the molecule in active site and make it more likely to change into product. یو ځل چېرته انزایم چینج کی slightly change دغه شي اکر شي distorting the molecule in active site او دا پکې وردن شي active sites ته and make it more likely to change into product او دې ته جوړ کی چې دا product ته convert شي. For example ورکړي if a bond in the substrate is to be broken that bond might be stretched by the enzyme. چې د سبسټریټ او د انزایم په منځ کې سبسټریټ بانډ جوړ شي نو ډیټ might be stretched by the enzyme کې دی شي هغه بانډ انزایم مات کړي making it more likely to break Alternatively, the enzyme can make the local condition inside the active site quite different from those outside, such as pH, water concentration, so that the reaction is most likely to happen. دوی به دغه خبره کوي منع کو چې د سپیسیفیک جیومټري ونه لاکن کیو کو سپیسیفیک نو سلایډي چین و بګیو شو انډیوس پټ ماډل په پالوږي صحیح ده ها بدی کې دومره سنشته ها دوی الټرنیټیولی د انزایم کین میک د لوکل کنډیشن انساید پی ایچ می ایچ هم چینج کولی شي د لوکل کنډیشنز quite different from those outsides چې هغه سره د outside نه different وي مطلب پی ایچ او واټر کنسنټریشن so that the reaction is more likely to happen بیا ما سره ریاکشن دي ته جوړ شي چې دا ما سره وشي اوس دا ما سره انرژي ها انرژي ده the way enzyme work also considering the energy change that take place during a chemical reaction, we shall consider a reaction where the product has lower energy and substrate is high. So substrate naturally turn into product. In other words, the equilibrium lies in direction of product. Before it can change into product, the substrate must overcome an energy barrier called activation energy. The larger the activation energy, the slower the reaction will be. The slower the reaction will be because only a few substrate molecule will the large this inverse relation there will be chance have sufficient energy to overcome the activation energy barrier. Enzyme په دې مې پوه کړی enzyme dramatically reduce the activation energy of a reaction so the most molecule can easily get overcome the activation energy barrier and quickly turn into product. For example, H2O2 the hydrogen peroxide the activation energy is kg 86 kg per mole او دا چې کله catalyst پکې نه وي نو 1 kilo per mole that is kilo per mole or one. The activation energy is actually the energy required to form the transition state. So enzyme lower the transition state. دا یو بل definition چې هغه انرژي چې ما د پکار وي چې transition state جوړ کړي. So enzyme lower the activation energy by stabilizing the transition state and do this by changing the condition within the active site of the enzyme. Active site of the enzyme. A cofactor are atom group of molecules that join with enzyme alternating their shape and make them functional. دا مثلا کوفیکټر یو اټومي مالیکول وي چې دا مثلا انزایم سره اټیچ شي alternating their shape او دا غي شیپ بدل کی او make them functional او هغه functional جوړ کی one thing of these cofactor is on off the on off یا switch off activating an enzyme switch off activating of enzyme. If the cofactor is non-protein like metallic ion, zinc, copper, or iron is referred as prostatic. Our cofactor, some cofactor are some organic molecule like the coenzyme. Prostatic group non-metallic. We like enzyme, they are not permanently alternate in the many of the coenzyme are derived from vitamins. Coenzyme must the vitamins or mineral navy. They are essential for life that the life the part really the absence of these cofactor can lead to vitamin and mineral deficiency. A lack of vitamin B produce that's usually berry vitamin B they can vitamin D berry दिल की बीमारी example of coenzyme are NAD FAD or NADP deficiency of vitamin B cause beriberi de कुछ شيء دي beriberi ber beriberi de beriberi beriberi برابری 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 برابر بر beri terasangkai enzyme nomenclature many enzyme but not all end in the suffix s exception pepsin or trypsin the name they name par the substrate they act on the mi pui kړی on the actions are performed پهله به دا کار و چې صرف ASE به دې ورسره واچو بس تا سره به د انزایم دغه جوړ شي لکه لایفیز یوریز مالټیز رایبونیوکلیز ایټیفیز داسې شي دي ډیکسټریز پروټیز ډیکسټریز the energy per lyphases take from ATP. Huh. Many but not oxidoreductase. These enzyme catalyze various types of oxidation reactions. Subclasses of group contain oxidase, oxygenase, and peroxidase. These enzyme catalyze reactions that involve the transfer of group from one molecule to another. Example of such group include amino acid, carboxylic, methyl, and carbonyl. Transcarboxylase and transmethylase are example of transferase. Methylase and carboxylase. Hydrolysis. The enzyme catalyzes the reaction in which the cleavage of bond is accomplished by addition of water. The hydrolysis include the esterase, phosphatase, and peptidase. Lysis catalyzes reaction in which group H2O, CO2 are removed to form double bond or added to a double bond. Decarboxylate, deaminase, or synthase are example of lysis synthase. This is a heterogeneous group of enzymes which catalyze several types of intermolecular rearrangement, ephemerase or mutase. Huh. Ephemerase or mutase. Ligases catalyze bond formation between two substrates. The energy for these reaction is always supplied by ATP. Hypothesis. Her temperature work best in optimum temperature. Enzyme work best in optimum temperature. Enzyme present in mammal work 40 animal present in different environment are adapt to range of temperature. For example, enzyme of Arctic snow da masara -10 the where in thermophilic bacteria work 70 up to 90 centigrade temperature the up to the optimum temperature the rate increase geometrically with the rate geometrically up to optimum temperature the rate increase geometrically with the temperature. It's a curve, not a straight line. The rate increase because the enzyme and substrate molecule both have kinetic energy. So collide more often and also because more molecules have reduced the activation energy. The increase in rate with the temperature can be quantified as coefficient which is the relative increase per a 10% rise in temperature. پوی کړی مې ای ها ټول ما خبرې دي. The rate is not zero at 0 centigrade, so enzymes still work in the refrigerator. صحیح ده؟ But they work slowly. خو صرف work. Enzyme can even work in ice through the rate of extreme slow due to very slow diffusion of enzyme and substance molecule through the ice lattice. Papain enzyme is found naturally in papayan. Papain enzyme papayan کې ملاویږي. Pepsin head او pepsin چې کوم دی دا مثلا پاپایان چې کوم دی یا پاپایان یا فیفین دی او پاپایان کې ملاوي دا په نیوټرل ټمپریچر papain had an optimum activity at widely range of pH 7 nanometer. The enzyme is able to break down organic molecule made of amino acid known as polypeptidase and plays a crucial role in industrial uses such as meat tenderizer pharmaceutical industry. Enzyme have دا مثلا ګوره د ډینیچر دی دا مثلا انکټیو دلته اکټیویټ شوی دی ورو ورو زیاتیږي دلته به کمیږي دلته مثلا ډینیچر شوی دی Alamasra influence of temperature on the rate of enzyme have an optimum pH at which they work faster. For example, this is about pH 7 to 8 physiological pH of most cells, but a few enzymes can work at extreme pH, such as protease enzyme in animal stomach. Huh. For most, the masculine sadna 8 physiological pH of most cells. But a few enzymes can work at extremes, such as protease enzyme, animal enzyme, which have an optimum pH. Protease of animal stomach pH only. The pH effect of change amino acid at active site must be change in pH ionized amino acid forming an enzyme separate can be longer one. As the enzyme concentration increases, the rate of the reaction increases nearly because there are more enzyme molecules available to catalyze reaction at high enzyme concentration. The substrate concentration more become rate limit, so the rate stop increase. The rate stop increase. The rate of reaction catalyze reaction slowly. The rate of an enzyme catalyze reaction. The rate of enzyme catalyze reaction slowly show occur depend on substrate concentration. As the substrate concentration increase, the rate increase because more substrate molecule collide with enzyme, so more reaction will take place. At high concentration, the enzyme molecule becomes saturated with substrate, so there are a few free enzyme molecule. So adding more substrate doesn't make much difference. Doesn't make any independent difference. Yeah. The rate of an enzyme catalyzed reaction slow show occur dependence on substrate concentration. As the substrate concentration increase, the rate increase in a direct way because more concentration is available can collide with enzyme molecule. सही ده यानी मतलब था रोटी सेवा की तो तखवगे खुराकम सेवा की ये तो काना है जल्दी का 1.5 multiplication pH से 1.7 trypsin 7 na 8 او یوریز کټلیز پاپاین دا هم د ټول په نیوټر باندې دي صحیح شو او لیفیز کې اډ لیفیز پنکریاس کې اډ دی په 45 61 68 مالټیز 61 68 inhibit the activities of enzyme, reducing the rate of reaction. They are found naturally but are also used artificially as drugs, pesticides, and research tools. There are two kinds of a competitive inhibitor or non-competitive has similar structure to a normal substrate molecule and it can fit into active site of the enzyme. It therefore competes with the substrate by the active site, so the reaction is slow. Sulfonamide sulfonamide in bacterial duct which acts as a sulfonamide in bacterial duct. And an incompetitive inhibitor is a quite different in structure from the separate molecule, doesn't fit in the active site. It binds to other part of enzyme molecule, changing the shape of changing the shape of changing the shape of changing, changing the shape of has happened changing relation inhibitor is reversible. It's quite different than it's bind to other parts of the enzyme, changing the shape of the whole enzyme, including the substrate molecule. Inhibit that bind weakly and can be removed out easily, or sometimes are reversible, while those that cannot be removed out are called irreversible. poison like cyanide, heavy metal, and some insecticides, and then computed cyanide of heavy metal. Feedback away. Another kind of inhibition is called feedback inhibition. In feedback inhibition, there is a second binding site on the enzyme where inhibitor bind. Feedback inhibition की वजह से कि ये the enzyme सर active site भी या binding site भी by one inhibitor bind she, so that the inhibitor is not necessarily similar in structure to the substrates only. The absence or presence of the inhibitor at the second site activates or deactivates the enzyme by changing the conformation of enzyme so that the active site is made available or unavailable to the substrate. This inhibitor is usually the product of reaction formed during metabolic. دي دا خبره کوي کنه چې ما سره فیډبیک میکانیزم کې داسې کار کیږي چې دلته ما سره په انزایم کې یو دویم اکټیو سایډ یا باینډینګ سایډ وي چې به یې وانې ما سره دغه انهبیټر بایند شي او دا inhibitor similar in structure to substrate we can in absence of inhibitor at second site enzyme activate or deactivate kulavi go bandi see that by changing the configuration of the sort active site is formed see that during metabolic pathway the the activities the activities some of enzyme is controlled by certain molecule binding to a specific regulatory site on the enzyme distant from the active site. Different molecules can inhibit an activity the enzyme, allowing sophisticated control of the rate. The sophisticated only a few enzymes can do this, and they are often a start of long bicarbonate pathway. They are generally activated by the substrate of the pathway and inhibited by the product of pathway, and thus only turning the pathway or when it is needed. Then pas masara proenzyme the holoenzyme the or iso proenzyme the holoenzyme masara the inactive form की holoenzyme masara natural active enzyme की भी pro masara both भी. No, Allah mu कामयाब को वस्सलाम.